Hello students. So today let's start with a new chapter, that is chapter 4 of class 11, Animal Kingdom. So first of all, in Animal Kingdom, let's talk about the basis of classification. So all of us know animals are multicellular heterotrophs. When we use the word animals, we do not include unicellular organisms. We generally include the multicellular organisms. And all the animals are heterotrophs and they don't have any cell wall present in their cell. Right? So when we classify them, animals are quite diverse. They are present in different habitat. They have different size, shape, structure. To study them systematically, to make them their studies very systematic and easy, we classify, we put them in the different groups. For grouping of organisms, we know that we have to decide certain basis. The scientist has classified organisms into different groups like Porifera, Coelenterata, Cnidaria, Ctenophora, so on. So these are classified on basis of these major 6 bases. So when we talk about these bases, the first basis is level of organization, body symmetry, germ layers. So that way we use word diploplastic and triploplastic. Then coelom or body cavity, presence of coelom or absence of coelom, their body segmentation, and notochord. So let's take one by one. So first, when we talk about the level of organization, right? So different organisms have different level of organization. Some animals have very simple body. As we know, animals are always multicellular. So these multicellular organisms may consist of simply cell aggregates, like in case of Porifera. They don't have tissue, right? So level of organization may include when they are consist of only cells, then we use word cellular level of organization, and cellular level of organization exclusively found in the Porifera. Porifera we will discuss in detail later on. These are commonly known as sponges. They do not have tissue. They are consist of cells. Individual cells are differentiated and they perform different functions. So they are present as the cell aggregates. Next level of organization we have, that is observed in higher organism than Porifera, is tissue level of organization. So cells come together that make the tissue. So tissue level means they have only tissues, no organs, right? Tissue level of organization, like it is present in case of coelenterate. We take example like Hydra. So Hydra do not have any organ, they have tissues. Please remember coelenterates like Hydra. Higher then we have organisms which have organ level of organization. Different tissues come together and that make the organ level of organization. So like member of Annelida, like member of Platyhelminthes, SK helminthes, they have organs. So like tapeworm, okay, tapeworm or member of Platyhelminths have the organ level of organization. Like us, they don't have organ system. We have respiratory system, we have digestive system, where many organs work in coordination and perform particular function. Then we use word, the highest level of organization of the body seen is organ system level of organization. Organ system, which is found in the higher animals. Organ system level of organization, like any example you can take, it may be like insects, arthropods, and so on. All these organisms have organ system level of organization. So if we talk about molluscas, arthropods, echinoderms, and all remaining group of animals have organ systems in their body. So this is the highest level of body organization. These organisms come under primitive organisms, and most primitive is Porifera because it have most simple level of organization. Life functions are carried out by cells, cellular level of organization. So whenever we classify, we always consider the level of body organization. Next we have is the body symmetry. So different organisms, if we observe carefully, have different type of body symmetry. Some organisms are symmetrical. Symmetrical means that can be cut into two equal halves. Their body can be divided into two equal halves from any plane. Then these are called symmetrical. And there are organisms which cannot be divided into two equal halves from any plane. Then these are called asymmetrical. So broadly, organisms which can be divided into asymmetrical and symmetrical. Right? Asymmetrical example, please remember, again we have to take the same example, sponges or porifera. So if we look into their body is like, okay, they are attached to the substratum. So you cannot divide this body into two equal halves from any of the plane. Their body are completely asymmetrical. So these are called asymmetrical organisms. Symmetrical again have two types of symmetry. This is called, most of the animals have bilateral symmetry, right? And another one is radial. Radial means either they are tubular or they are spherical. Okay, so like organism is spherical, so it is its axis, so it can be cut into two equal halves. Okay, if we cut it from the axis, the organism can be divided into two equal halves from any plane. Then this type of symmetry called radial symmetry, or this is seen in Hydra. Tubular organism is there, then also it can show the radial symmetry, but condition is the cut must pass from the axis, then it can be cut into two equal halves. There are organisms like including human body. Our body can be cut into two equal halves from a single plane. That is, from both these sides are called lateral sides. So from can be cut into two equal halves from single plane. Then they are called bilaterally symmetrical, means they can be divided into two parts. Only bilaterally they can be divided. So please remember, organism can be asymmetrical. Most of the animals are bilaterally symmetrical, but some organisms like Hydra or member of Coelenterate, which are spherical or which are cylindrical, they are radially symmetrical. So symmetry is very important basis of classification of animals. Third basis used for the classification of animal is germ layers, or we use word organism is diploblastic and triploblastic. Germ layers develop when zygote divide and that develop into different stages like blastula, gastrula. So during gastrulation there is formation of layers in embryonic development. These are called embryonic layers. These are called germinal layer or embryonic layer, which are formed during development of embryo. So in lower organism, these germinal layers are only two. These are called diploblastic. When three cell layers are found in the developing embryo, or three germinal layers are found, then it is called triploblastic. So please keep in mind, diploblastic and another one is called triploblastic. Triplo means three. So embryonic layers, we use word germ layer, or these are nothing but the embryonic layer. Layers present in gastrula, early embryo. Embryonic layers. So we can understand by drawing the diagram. This is the one layer and this one is another layer. So in between two layers there is a non-cellular. So as I mentioned, these layers are cellular. This is called ectoderm, and inner is called endoderm, right? In between non-cellular substances present, that is called mesoglia. This substance is called mesoglia. Ectoderm, derm means skin, ectoderm, endoderm, this is the lumen, that is can be gut, lumen, right? And in between these two layers, non-cellular, this mesoglia, please remember, is non-cellular. This is not consist of cell. Mesoglia is present. This is non-cellular thing is present. This is the diagram showing the diploblastic. This kind of organization is present in the member of coelentrates, platyhelminthes, which we generally consider as lower animals, right? Then when we talk about triploblastic, beside ecto and endoderm, there is presence of one more layer, and that is called mesoderm. So outermost is ectoderm, mesoderm and endoderm. In early embryo, 3 embryonic layers are present, then we use word triploblastic, and 2 embryonic layers are present, then these are called diploblastic. All higher animals are triploblastic, and from these germinal layer, cellular layer, after differentiation during embryonic development, different organs develop, like from ectoderm body wall develop, from endoderm intestinal wall develop. Similarly, internal visceral organ may develop from the mesoderm. So on basis of embryonic layer, we can say these are diploblastic and triploblastic. Then we have another basis, that is called coelom. Sometimes we use word body cavity, but please remember body cavity. So all the organisms have a body cavity, but here coelom term stand for the cavity, right? So if we consider, this is body wall, and this one is gut, intestine. So this gap which is present between the lining of outer lining of the intestine or gut and inner lining of body wall. So the gap present between the inner lining of the body wall, right, and outer lining of the gut is known as coelom or body cavity. Please remember, this is called coelom. This cavity, which is intestinal cavity, is not considered as the coelom. So coelom is the gap or the space present between the inner lining of the body wall and outer lining of the intestine. So on basis of the presence or absence of cavity, we classify animals into 3 categories. So animals which have true cavity means like this, tube within tube. So this body plan is called tube within tube. This is a tube, and inside the tube is present. In between two tube, whatever gap is present, fluid filled gap may be there, or their gap is known as that, like in case of insect, okay, that is fluid filled. Then we use word hemocoel, okay, and that is, these animals are called coelomate. When true cavity is present, word used is the coelomate. Okay. Now if this body cavity is present, but this is vacuolated. Vacuolated means most of the cells present, but in between here some space is present. It means there is no true cavity present. Okay, most of the space is what we can say filled, and in between some space is present. Then we use word pseudocoelomate. Pseudocoelomate, right? This is called false cavity. And false cavity, we need to remember only one example is there, and that is very important example. Member of Nematoda or SK helminthes. This is the only group which have SK helminths. Please remember, or nematodes. This is the animal group which have the pseudocoelomate, false body cavity, false coelom, right? And there are organisms like member of platyhelminthes or flatworm, or then lower organisms like coelenterate, porifera. They do not have body cavity, right? It means they do not have gap between intestine and the body wall. Okay, then they are called acoelomate. These are called acoelomate. They are like this, right? So this is their body lining. Okay, and here there is no another tube. There is no cavity present between intestine and body wall. Then these are called acoelomate. The term used is acoelomate, right? Like member of platyhelminth. Platyhelminth, coelentrates, these are acoelomate, lower organisms. So very simple way to remember is, when we continuously talk about like Porifera, Coelentrata, okay, Ctenophora, Platyhelminthes. So till Platyhelminthes, always remember acoelomate. Then next group is Aschelminthes or Nematoda, pseudocoelomate. And after Aschelminthes, Annelida and onwards, all animal groups are coelomate. So before Aschelminthes, all primitive groups are acoelomate. Aschelminthes, pseudocoelomate, and after Aschelminthes, all rest of the groups are called coelomate, means they have true body cavity. Next basis used for classification of animals, that is the segmentation. What we use segmentation? Segment term itself is self-explanatory, that division in the body. So organisms may have internal division. So when organism is truly divided, there is a compartmentation inside. Like we take best example, earthworm. Okay, when you look into the body of earthworm is not only externally segmented, that is internally also segmented, right? If organism is internally segmented and there is formation of these compartments, then organism is called segmented. This kind of body is called segmented, right? This type of segmentation is called true segmentation. Like our body have thoracic cavity and abdominal cavity. Similarly, like insect are divided into head, thorax and abdomen. So these bodies are segmented. These animals are truly segmented animals. Okay, so we take example of earthworm. Here repeated segmentation is there. So if truly segmented and this segmentation is repeated, means one or another part is similar in all these segments. Okay, then this type of segmentation is called metameric segmentation. Repeated segmentation is called metameric segmentation, right? This best example is the member of Annelida, metameric segmentation. Each segment is called metamere. Metamere. And if body is not divided into these segments, then this is called asegmented. Okay, body is not divided into segments. So please remember the very important term, when body is truly segmented and these segmentation are present repeatedly. Internal segmentation is repeatedly present. This repeated segmentation is called metameric segmentation, and each segment is called metamere, right? Last, we can take notochord. So if you observe animals, in some animals generally we observe that there is no presence of backbone. Okay, notochord is a rod-like structure. Rod-like structure. Okay, it is always present in the dorsal side of the body. Dorsal side means back side of the body, right? So if notochord present, then these are called chordates. And if notochord absent, then these are called non-chordates. That is very simple word. They are called non-chordates. So we classify animals on basis of presence or absence of notochord. Higher animals, the last phylum that is having the presence of backbone, this rod-like structure in the dorsal part of the body, then these are called chordates. In some higher animals, okay, this rod-like structure, that is notochord, is replaced by vertebral column. When it is replaced by vertebral column, then please remember these are called vertebrates. Vertebrates. So when notochord is replaced by vertebral column, then these are called vertebrates. In lower chordates like protochordates, hemichordates, there may be presence of this rod-like structure only, and this rod-like structure may be persistent for whole life, or that may be present in the early embryonic stage, or that present in only adult stage, that present in the complete body, that may present in only tail region. Okay, likewise. So these are the basic characters, basic features we use to classify the. Or beside that, some organism may have open circulatory system, some have closed circulatory system. There may be presence of exoskeleton, presence of endoskeleton, aggregate cellular body structure or tube within tube body structure. Beside that, we use some other basis also for the classification. So that's enough for today. Thank you.